looking at human genetics and epigenetics with chapters 2 and chapters 3. I realize it is a very long read on some of these chapters and on the books. What I would recommend to you, just for overall success, is skim the chapter summaries, if anything. Skim the chapter summaries. Read through those. If you want to understand a little bit more of the content, definitely read those components of the chapter. But at the very least, just kind of gain some insight by looking at the course summaries. Also, looking at the lecture content ahead of time just to see if it matches with some of the reading content. We won't cover every single bit of those chapters, but we'll cover a good amount of it. And then also just follow along with the weekly lectures, and that'll help prepare you at least for the content to expect on the quizzes. I also have one more thing that I'll show you here as we wrap up the session, which I think you'll find a bit useful as well. So human genetics is our first one. All right, so whenever we're looking at genetics, ultimately we're looking at the study of inherited traits. And there's a ton of variations that we can likely see depending on, again, the material passed on from maternal and paternal sources. Genetics itself is a life science, not related directly to genealogy, which is another topic. What would genealogy be, by the way, guys?
The study of genes.
Genealogy. Yes, in the sense genetics, there's a bit of a difference on that versus genealogy. And I guess it's looking at a different aspect of kind of the human makeup of sorts.
Is it who's related to who?
Yeah, in a sense. Yeah, and more so in a sense. But not necessarily looking at a generalized makeup of sorts. All right, genes. I'm going to bring up, here we go, all of the little points there. As far as genes go, we want to think of them as a unit of heredity. So we're getting it from something. Mom's giving it to us, dad's giving it to us, maybe they're both giving it to us. And these could be very selective based on, again, just a combination of variations that are passed down to us from maternal and paternal sources. So ultimately, they make up these biochemical instructions and really tell the cell how to manufacture certain proteins. What we'll learn is that the body is a protein-making factory, and these proteins, as small as they are, have very encrypted codes, very specialized codes in terms of how they should function. Thousands of these proteins that have some form of instruction and very specific, manufactured combination in order to give us some type of instruction. These proteins could be related to maybe brain tissue. They could be related to the liver, the bone. They could be related to how we look. All types of different instructions and directions are derived from these proteins. They consist of DNA, or deoxyribonucleic acid. And with DNA, what we'll find is the double helix structure. And the double helix structure of DNA helps to combine a set of different amino acids. Within amino acids, pairing different amino acid combinations gives us the ability to form a protein. But ultimately, again, genes will consist of DNA. The traits interact between genes and their environment. Their environment can be manipulated in a variety of ways. We'll explore a little bit on how that is possible. The genome is basically a complete set of genetic instructions, so that it forms an organism, which is us. So we're the human genome, we could say. We have a ton of different instructions within our cellular makeup. And we we are all unique in many ways and very much so similar in many ways, for that sense. Again, the instruction that comes to us and helps build who we are is really encrypted and encoded in very specific proteins that have instructions for all types of different functioning abilities within our body. There are varying DNA sequences for these particular proteins. Genomics, if we're looking into genomics, it's a field that looks and compares genomes of different species. It could be same species, but it could also be of different species as well. This is when you get into conversations about how much we, as mammals, share traits with other mammals, we could say. How are we related to dolphins? How are we related to primates? That kind of thing. But also comparing human and human. So there definitely falls importance in the study of, again, how life is related and intertwined with one another. We have varying levels of what we know of as genetic levels, and really it starts at the microscopic level. What we have is the cell, and within the cell we have a nucleus. So here pictured to the left we have the cell and its cell wall. Usually these are kind of more of a spherical shape of sorts, although cells are a little bit more flexible in their shape based on things that can enter and exit freely. Mostly like water. Water can easily move throughout membranes, intracellular and extracellular areas. So that could also that could contribute to the form and shape of a cell. But a lot of the cells share a lot of similarities, in which would be the components that make them up. So we have mitochondria. These would be like our protein-making factories of sorts. The ribosomes are the protein-making factory. The mitochondria are kind of like the lungs of sorts of the cell. And then we have the nucleus. So the nucleus we see kind of like in a half-dome sense of things toward the bottom of the page. And it's got chromosomes. Those chromosomes on a microscopic level break up even further into what we would distinguish as genes, which is a bundle of proteins that are set and created to form particular instructions for our body. And if we unravel that further, then it becomes to the point where now we're looking at DNA, the double helix, the combination of amino acids and its pairings. So there are very deep levels to this, and mostly when we're looking at some of the deepest levels, we're looking at DNA and its sequences, because those sequences are specially formulated, again, to form proteins, and those proteins in bundles we collectively know as genes. Those hundreds, thousands of genes make up the material that is found in chromosomes, and it's kind of like a blueprint of sorts of what we are as humans and how we are likely going to develop. As far as the human genome, we have 23 chromosome pairs. The 23rd pair it's a little bit small to see, but it's the XY or XX gene. This could vary as well. There could be some extra components to the sex chromosome component, in which there could be differentiating sexual characteristics that could emerge based on what's going on. For example, there could be XXY, or maybe we're dealing with single genes, so it might be just an X. So depending on what's going on, we wouldn't say male-female under traditional standards. We would say there might be a syndrome that follows it versus listing male versus female. But under traditional components, we would say XY would be male, XX would be female. bioethics. This is something that you might run into later in the progression of your program, especially with the ethics course that you'll be taking, likely semester 6, unless you take it sooner, if the progression changes. But in terms of bioethics, we're looking at addressing moral issues or controversies related to medical technology manipulating our genes. And this is kind of what I'll kind of have you break out into some conversation. Let me go back here. Some conversation related to modern day concerns that you've noticed or you've heard about related to like the manipulation of genes. So this one's a free for all. What are some of the newer concerns that you're seeing nowadays as far as gene manipulation, or maybe using technology to alter certain genes?
Like studies about choosing male or female for your child.
Okay.
So I had to go through IVF quite a bit, and there was a lot of stuff that has to do with that as well. Making the blastocysts and all that stuff. And a lot of people actually do IVF for reasons of genetic issues to try to prevent them from passing on those specific genes to their child.
Yeah. So even with those two examples, there's an expanded conversation that could be had with those. And if we're looking at being able to choose gender, that's one thing. If we're looking at the use of technology to possibly prevent some genetic instructions that have not been as helpful in health processes, that could be another thing, and possibly beneficial too. Would you guys think that this technology would be used a little bit frivolously if it was perfected to the point where you wanted to have the child, let's say, you can choose exactly how they look. That could be color of eyes, height,
I think
gender.
that's a little extreme. Most people that are doing this type of thing don't necessarily care about eye color and hair color and stuff like that. They just want something that's healthy.
And
Yes, but I think but if I think that it was like you said perfected, and it was just a thing, like a common,
Like you can just go shop and pick what you
yeah,
think you
I
want.
bet you that it would definitely be. I mean, people would use it and take advantage of it, and I mean,
Well, for sure.
that would be scary
It
though.
would
I
be a
don't know.
whole lot of money. It's already $30,000 just for plain. Yeah.
I agree. You could build your little star athlete.
Right, there would be no like
You-
uniqueness in a sense.
Yeah.
Build. Yeah.
I like your conversation. So yeah, it's always kind of again something that could be controversial, but imagine a world that can perfect it, and that day's coming in our lifetime. So that's something to consider too.
I'm pretty sure it's there if they can pick and choose certain genes and stuff already for certain disease processes and whatnot, male, female. I'm sure it's already in the works. We just probably aren't aware, or it's not mainstream.
Yeah, definitely, and and I guess a spin-off the topic to that would be kind of related to other potential reasons. Some folks might have a second child or a third child, and that again has been a controversial concept in the past, but also to just kind of mention the child would be basically born to help the other child. Have you guys heard of that particular topic?
Yeah, there's actually a movie on that, My Sister's Keeper.
Yeah, My Sister's Keeper was kind of addressed that controversy in some sense, the legalities behind it. But since they're coming up with different technologies too, there's also talks of future 3D printing of organs. So that would have to be something that could hopefully, if they perfect that kind of stuff, I don't know. I might run out of hair and might not be able to see at that point if that's perfected. But there's definitely technology that's emerging that can help solve some of these potential issues with genetic, with all types of genetic issues. All right, I'd like your input in this, guys. DNA, deoxyribonucleic acid. For testing purposes, I do want you to know its composition. So let's take a look here. We have different components. The phosphate group, 4 nitrogenous bases, basically protein bases. These are more or less your amino acids: adenine, thymine, cytosine, and guanine. And then there's a sugar group too. Phosphate sugar, and then your nitrogenous bases, which are basically like amino acids. So each consecutive 3 DNA bases is a code for a particular, let me go back to this, a particular amino acid. So it would be kind of like combining A, T, C, or G in some type of triad. Maybe you have thymine, cytosine, guanine. And then in another set, you have adenine, guanine, cytosine. So those particular 3 bases create a code for a particular amino acid. There's a long list of amino acid combinations that can emerge, and you guys don't have to know that in particular for the scope of this particular course. If you ever go on for advanced pharmacology, that'd be something that we would review at a different time.
I have a question.
DNA double helix.
I have a question really quick.
Yes, question.
When the bases, so will it always be 3 combined together, just 3 of them? Or will it like differentiate?
It would be like a set of 3 in a set to create a particular amino acid. So if I wanted a particular amino acid, it would be kind of like a very similar or same sequence of sorts.
Okay. Thank you.
Yeah, no problem. But double helix looks at those particular 4 amino acids that we discussed. So we're looking at guanine, cytosine, thymine, and adenine. So we have some that combine with one another in kind of groups of sorts. So we could say A and T, or adenine and thymine, have an affinity toward each other. So they'll combine with one another. Cytosine and guanine will combine with one another. So take a look at this little chart here and kind of notice the groupings. If you start with T, it'll combine with A. If you start with A, it'll combine with T. And same with cytosine and guanine. C matches with G, G matches with C. And that would be another thing I want you to recall for testing purposes. In fact, I'm going to give you a quiz question that will very much so relate to the content that you'll see on the test. So I'll give you a random this combination. You see my annotations? Can you see the writing on the red writing on the slide?
Yes.
Yeah, okay. Again, I'm just doing a random selection of some of these particular setups here. All right, so let's say this is my base. I'm not particularly making any type of amino acid. I'm just creating a double helix here. What would be the complementary strand to finish off my double helix here? I have a single strand now. But what would complement this particular strand?
You want us to type it out or say it?
No, you can go ahead and say it out loud.
So it would.
A G T
So,
underneath. Is that what you're saying? Go underneath it, Professor, or?
yeah, so it'd be going underneath it. So remember, G matches with C and vice versa. T
So would
matches
it
with
be-
A.
C under the T, then A, G, T,
A.
G and C, right?
and then G, C, G, and then C, and then T.
Yeah. So for test purposes, I want you to be able to identify complementary pairs. And that'd literally be a question on the test. I'd say, what's the complementary strand for this? And under the concepts of the amino acid pairings, we would say C matches with G and vice versa. T matches with A and that vice versa. Just kind of, again, kind of do the opposite of what this other strand is doing.
I have a question. Would you ask like 3 to 5? Like the correct order would be TC instead of CA versus 3 to 5? Or no? Is that too? I was wondering. I've seen this kind of questions.
Those types of questions.
Like, because it would be 3 to 5 and then 5 to 3.
I won't necessarily go as deep as asking that particularly. I'll just in this same format just kind of give you a strand, and then you'll give me a complementary strand.
Okay, perfect.
All right, so that's the double helix. And again, get the complementary to the base is going to be ideal to give this its full function. Now, if this was a single strand, we'd be looking at ribonucleic acid, so RNA. Anytime we're dealing with cell division or a replication of sorts, we're going to look to RNA to kind of split off from the double helix. And what it ends up doing is kind of cloning a particular segment of the DNA strand to form, well, ultimately new proteins. But with RNA, it goes through a process called transcription. There's usually within the double helix some type of starter protein and a protein that says stop. And what I mean by that, I'll go back to the other slide. There's some type of start and stop, and I'll draw it on annotation. So let's say my protein now says, all right, we're going to start here, and we're going to take this entire strand up to about here. And so everything along the line, like C, A, G, T, C, T, everything that you see there is kind of taken out. And you have a single strand at that point. And then another amino acid comes in and fills the gap. So another amino acid comes in and fills the gap to close out that helix. And these are just kind of free-floating, realistically. Kind of putting together a puzzle of sorts. But the RNA strand would be taking a strand of that double helix and making use of it in terms of creating replicating of sorts the DNA sequence. So you're taking copies of one strand of DNA, and then at that particular point, trying to find a complementary pair. But this is now going to be the end goal of this is going to be to create a particular amino acid. So when it does break up, it's going to break up into the sets of 3 of sorts. So I guess my amino acid would be something like this. So it'd be kind of like a amino acid that's formed, and a complementary amino acid would follow, except when we're looking to complement RNA strand, we would replace T with uracil, which would be a U. I'm not going to have you, for the sake of testing purposes, give me combination pairings for amino acids. I'm just going to have you give me a complementary strand for DNA, as we did in the previous example. but just to kind of give you again a notion of RNA, it's basically a little sequence of the strand of DNA. And we're looking to create amino acids with them. The amino acids ultimately align and form a protein, and that protein gives us direction, depending on, again, where this DNA came from, which part of the body. So in order to really have replication of our DNA, we have to take little segments of it, kind of make copies, and be able to form specialized proteins based off of these RNA strands. A little bit more about the human genome, which is us. Only about 1.5% of our DNA actually encodes protein, but we have a whole lot of protein encoding genes, over 20,000, and this means that we have 20,000 plus set of instructions that allow our body to function as it does. There are some genes, though, that are known to cause disorders and different traits, and these are cataloged in a database that is used globally. So this one's known as the Online Mendelian Inheritance in Man database. What's important about documenting this, guys? Would it be a good idea to have some type of documentation that certain syndromes and traits are occurring? It should be, right? And if globally, since we're a human genome, this could happen anywhere, and we could learn from each other from these particular issues that may arise. Chat box. Let's see. They use DNA for cancer treatments. Are you? The question was, I think it's a question. Is it a question or a statement?
No, I was just saying that, like, they take your DNA now when they're doing they're doing your chemotherapy, to make sure that the chemo that you're using will work with the cancer, right? So that would be important for them to know what our DNA is, you know.
You're talking about like a bone marrow transplant type thing?
No. So when I had a friend that had liver cancer, so they took her DNA to make sure that the cancer would work with her body and the cancer for her treatment.
Oh, I see.
So like the City of Hope actually takes your DNA to make sure that how they're treating you is going to work for your body specifically, which is pretty cool.
It's kind of like testing
before you actually get the treatment. Yeah, exactly.
Yeah.
It's pretty outstanding.
There was another question about PowerPoint slides being available to print. I wouldn't recommend printing this, guys. It is under the file section. All the PowerPoints that we'll be using this term. If you need it tangibly in front of you, if you want to print it, it's up to you. But since it's online, it could be accessible to you on mobile devices, on your computers. So it's kind of up to you how you want to see these. But back to the cancer comment. Yeah, that's pretty neat how they would be able to, again, before giving a particular administration of chemotherapy agents, seeing if it would be effective, instead of giving it to somebody and potentially it causing no real beneficial effect and more detrimental and suffering of sorts. With chemotherapy agents, we'll be talking about cancer in a couple of weeks, but I'll kind of show you how sneaky cancer can be in terms of how it gets into the cell cycle and ultimately ends up spreading to other organs. All right, alleles. These are variation of genes, and what we want to know about alleles is because of the different variations of genes, this is something that can be a potential opening for mutations too. So changes in DNA sequences that distinguish alleles arise by mutation. And mutations in sperm or egg cells are passed on to the next generation. That means maternal and paternal mutations could be passed on to offspring. And these are some of the things we were kind of talking about during the short discussion on bioethics. If we could prevent this, should we? And to what degree? So those would be things related again to that particular conversations. But not all mutations are negative. There are some mutations that do take place that could be neutral, and sometimes they could be very helpful. Based on environmental changes, perhaps.
You're cute.
And
I said you're cute.
usually look at mutations as possibly more negative than not. But again, they could just be neutral, or they could also be positive. Chromosomes, as you might have recalled from the picture, the chromosomes were basically the set of instructions that were inside the nucleus. Kind of looked like an X shape on the illustration. But otherwise, these are what we're looking to as far as describing the different chromosome pairs. So we have 23 pairs of chromosomes, 46 chromosomes total, we can say, under normal circumstances. 22 pairs of autosomes. Those are going to be chromosomes 1 through 22. And the final set is going to be our sex differentiating chromosome, so to speak, which mostly would be XX or XY. So a pair of sex chromosomes would be the 23rd pair. And this kind of reiterates again, traditional labeling of female versus male. 2 X chromosomes would be female, X and Y would be male. And here it is illustrated. Now the karyotype chart, what we're looking at too to identify maybe certain disorders would be to see if there are any extra parts outside of the normal count that we would expect. So maybe we have an extra chromosome on chromosome. So I'll throw out a more popular one here. Maybe chromosome 21. That would give us indication that there's the presence of perhaps Down syndrome.